Today we're talking about Mendel's observations that he made during his experiments. We need to learn this to describe and understand how geneticists explain inheritance, and our success criteria are being able to identify the P, F1, and F2 generations, to differentiate between dominant and recessive alleles, and being able to differentiate between genotype and phenotype. So, Mendel's experiment was set up so that he broke it into 3 generations. We're going to do this example with pea plant height, but Mendel actually did it with all of the different traits that we saw in the last lecture. So if we're using height as an example, Mendel's first generation, or the parental or P generation, would have been a purebred tall plant and a purebred short plant. What purebred means in this context is that if you breed it with another plant of the same type, it will produce the same type of appearance. So the tall plants will produce tall plants, the short plants will produce short plants, and only that type of plant. From the offspring of the tall plant and the short plant, what Mendel saw was that all of the plants were tall plants in the first offspring generation. We call this the F1 generation. The F stands for filial, which just means children. Okay? So the F1 generation, the first generation of offspring, were all tall plants. Then, because pea plants can self-pollinate, what Mendel did is he got the tall F1 plants to self-pollinate. So they were matching their own DNA against itself to see what that would do. And the reason that Mendel did this is he was making sure that the same traits were being used, that exactly the same traits were being used this time. And what he observed was an F2 generation, a second generation of offspring, grandchildren of the original short plant and the tall plant, and he saw that there was a 3-to-1 ratio in that generation. Unlike the F1 generation where everyone was tall, in the F2 generation, there were an average of 3 tall plants for every short plant. And this is something that's very strange and a little bit unexpected unless you understand the patterns of inheritance that are driving these traits. So what Mendel discovered out of this was that every single plant must have 2 copies of each trait. The short trait was still present in the F1 generation. It was just being hidden by the tall trait. Mendel called these traits factors. We now know that these factors were actually the alleles on the gene that causes the plant to be tall or short. So I'm just going to call them alleles from now on. What Mendel was able to see was that the alleles were separating independently in the F1 generation and in the F2 and in the P generation as well. We call this the law of segregation. Gametes are going to pass on alleles randomly, and it's going to be one of the 2 alleles that the parents possess. Mendel also came to realize that the F1 generation had to have both the tall and the short alleles. They got 1 allele from each parent. The reason that the tall allele was the only one expressing, though, was because it's what we call a dominant trait. It will show up no matter what you pair it with. It will dominate other traits. The short allele, on the other hand, could only be expressed if it was paired with another short allele. So we call that a recessive trait. The recessive traits only show up if you match them up with the same type of trait. They are recessive to the dominant. We call this the principle of dominance. When individuals with different traits breed, the offspring will only show the dominant trait. So if you look in that bottom right corner with the F2 generation, we see that there are 3 tall plants. But one of those tall plants has both of the dominant alleles, and the other 2 have the dominant allele and the short allele. Notice, though, they're all the same height. It doesn't matter. There's no blending of those traits. The tall trait is completely overriding the short trait. So when we start to describe heredity, we need to differentiate between an organism's phenotype and an organism's genotype. An organism's phenotype is the trait that appears or gets expressed, because not all of these traits have physical appearances. So we can see that in the F2 generation, there are 3 plants that have the tall phenotype and 1 plant with a short phenotype. In the F1 generation, they all have the tall phenotype. But phenotype is different from genotype. The genotype is the organism's genetic makeup. And the way that we represent genotypes is we represent them with letters. We use capital letters to represent dominant traits. We use lowercase letters to represent recessive traits. So in that F1 generation where everyone is tall, all of the plants we would say are big T, little t. And you'll notice that we're using 2 T's, not a T and an S for tall and short. We always pick one letter, and you'll see why in a couple of lessons. So the tall in the plants is represented by a big T, the short in the plants is represented by a little t. All of the F1 generation is big T, little t. But when you cross the heterozygous, 2 different traits, with themselves, what you end up with a variety of possible combinations. You can get some plants that are big T, and will be tall. You'll get some plants that are big T, and their phenotype is also tall. And then you get other plants who are little t, which means they will have the short phenotype. Genotype is what determines an organism's phenotype. Okay? So if you have a big T at all, you will be tall, but if you have 2 little t's, you will be short. So an important thing to take out of this is that the genotype ratios are different from the phenotype ratios. The phenotype in the second step of Mendel's monohybrid cross, which is what we call this one trait observation, the second filial generation will have a 3:1 phenotypic ratio. In this case, it's flower color instead of height. But they will have a 1:2:1 genotypic ratio. The first one is the homozygous dominance. They have 2 copies of the dominant trait. The 2 is the heterozygotes, the individuals who have 2 different traits but are only expressing the dominant trait. And then the second one is the homozygous recessive individuals who have 2 of the same trait, but instead of the dominant trait, they have the recessive trait. And it's these homozygous recessives that are the only ones who express the recessive phenotype.